again, and I'm delighted to have the opportunity. And today I'm going to talk to you about what I see as a role for long-acting microneedle delivery systems in a post-COVID world. So I'm going to talk about the role of drug delivery in recovery of healthcare systems, and how to do this we will need to change our mindset to consider transdermal delivery of high doses and controlled release of medicines after we've actually removed the patch. To do this, there are a range of different design and manufacture considerations we must take into account. But I think that there are a number actually of microneedle platforms that can contribute in this area. Dissolving microneedles and swellable microneedles, of course, but also microneedles that form depots within the skin. And finally, in order to achieve these benefits for patients of at-home delivery of long-acting medicines, then there are a number of patient and translational considerations that we need to overcome. So obviously we've been very much aware over the past 19 months that the COVID-19 pandemic has had a profound and very detrimental effect on the normal functioning of healthcare systems worldwide. And because there has been such a prolonged effort at diagnosing and then treating in hospital, but also in community settings, COVID-19 patients, there has been a substantial backlog develop in the normal business of healthcare organizations in terms of diagnosis and treatment, particularly inpatient treatment, where people are getting surgeries or major diagnostic procedures. So routine injectable drugs still typically require attendance at a healthcare setting. So this could be something like a long-acting contraceptive injection, something for treatment of schizophrenia, some treatments for prostate cancer, but increasingly treatments for lifelong diseases like HIV and tuberculosis. And typically people will have to go to a healthcare setting, and this obviously adds significant cost and takes away time from healthcare organizations in developed countries. In developing countries, it creates an even bigger problem because there has already been a massive strain on those healthcare organizations even before the pandemic due to a lack of trained staff. And for patients, there's also the problem in developing countries that they may have to walk for 2 days to get to a healthcare setting to have an injection. If we then add to this with the ever increasing prevalence of injectable medicines, so there's a big drive at the moment in the pharmaceutical industry for development of long-acting therapeutics. And they aren't just small molecules, they can also be long-acting large molecules. So for example, glucagon-like peptides for treatment of type 2 diabetes, or therapeutic antibodies that would typically be administered on a weekly or monthly basis. Now, if we can take the delivery of these advanced medicines out of healthcare settings and put them into the hands of patients in their own homes, then we would really free up healthcare providers. So this will save money, allow people to start overcoming this big backlog, but also avoid risk of transmission of COVID from a person actually bringing COVID into a healthcare setting or requiring it while they're actually there. If we then consider that there are still considerable issues with needle phobia and inappropriate reuse and unsafe disposal of normal needle and syringe based injections, we can really see that basically transferring intramuscular or even subcutaneous injections into people's own homes is not necessarily the answer. And as we know, oral administration of most injectable drugs is not feasible or it's extremely inefficient. So if you take a glucagon-like peptide, so something like Zenatide orally, the bioavailability is probably about 1%. If you take a therapeutic antibody orally, it's probably even less than that. So that's not going to be commercially acceptable. And then if you've got a long-acting depot-forming system based on either nanocrystals of a poorly soluble molecule or a biodegradable polymer implant, then you can't really just take those orally, because they're probably not going to be absorbed. They're just going to pass out unchanged from the gut. So we need to think about alternatives to that. One alternative would be to deliver medicines across the skin. And we well know that the worldwide transdermal patch market is extremely valuable, despite being based on less than 20 drugs. And clearly, even though people have been working on this since the late 1960s, early 1970s, there still are a number of barriers that we simply can't overcome, because they're physicochemical barriers related to the stratum corneum and also the properties of drug substances. So if your molecule is too large or too hydrophilic, it's basically not going to penetrate across the stratum corneum in any sort of an efficient way, and you're not going to achieve therapeutic plasma levels. So what this means is that many drugs that we would want to deliver into or across the skin don't possess the necessary characteristics. And enhancement techniques like penetration enhancers, prodrugs, and ion pairs all have limited enhancement capacity. So they'll all give you a 1 order of magnitude increase in transdermal delivery, so about a 10-fold increase. And that's probably enough if you're almost there in terms of achieving a therapeutic plasma level over a period of time. But an order of magnitude greater than nothing is still nothing. So for biomolecules or particulates, then forget about using those techniques. The more advanced techniques like iontophoresis, sonophoresis, electroporation, and jet injection all actually do work with certain limitations, but they tend to be overly complicated or not very user-friendly. And in the case of iontophoresis, very often the products don't tend to be terribly stable, because the gel that is necessary as the drug reservoir tends to corrode metal and metal chloride electrodes. So, and then obviously these are expensive as well. So they all have limited impact. So we clearly need something that's more simple and easy to use by patients and can be produced at fairly low cost. And I think this is where microneedles very much come in. We know, though, that microneedles have a micron scale, so they're typically less than 1 millimeter. So they're not going to cause pain, they're not going to cause bleeding, but because of the relatively small height and the typical small patch size, the dose that we tend to be able to deliver from a normal coated or hollow or dissolving microneedle tends to be quite limited. So a lot of work obviously has very rightly focused on the advantages of microneedles in vaccine delivery, but I think that with appropriate formulation design, that there's real potential for drug delivery here. So we need to consider then that microneedles create aqueous pores in the skin. So if we've got particulates, particularly hydrophobic particulates, if we've got an implant made from a biodegradable polymer, then we need to think, how do we actually deliver something that's not terribly water soluble in suitable doses through these aqueous pores, particularly when we might need tens or hundreds of milligrams for a weekly or monthly or even daily dose in some cases. What implications would all of that have on the size of the patch we need to make? And then can a person apply such a large patch? And what happens if they repeatedly apply the patch to the skin? Because remember, for drug delivery, the microneedles are going to be applied on an ongoing basis. It's not going to be 1 or 2 applications as we would have with vaccination. In terms then of manufacture, we need to consider what the cost considerations will be. Can we be cost competitive with tablets or conventional injections? And I actually think all of these things can be readily overcome, and that this will create a range of competitive advantages for industry and in turn considerable benefits for patients. So if we're going to do this with dissolving microneedles, then we need to have a high loading of drugs. So the needles themselves need greater than 50% weight per weight of drug, because on a typical microneedle array, you might only have, say, 6 or 12 milligrams of needle weight per square centimeter. So we need to get a lot of drug per unit area. We're going to be depositing polymer in the person's skin here as the microneedles dissolve, as well as the drug. So we need to have either a biodegradable polymer that the body has the molecular machinery to chop up and excrete, Or if we're using a typical carbon-carbon backbone pharmapolymer like polyvinyl alcohol or polyvinyl pyrrolidone, then we need to use a low molecular weight, less than about 60 kilodaltons, so the body can readily clear it by glomerular filtration. We will be casting these gels containing our drug at a high loading into a mold, and we'll be drying. So the polymer has to be a good film former, and ideally we should have also a low glass transition, because with a high drug content, we're going to need something that forms a very nice homogeneous structure that also is still quite hard to penetrate the stratum corneum. Ideally, we'll dry slowly to promote physical stability. The last thing we want is to trap the drug temporarily in an amorphous state and then have it slowly recrystallize over time, because that's clearly going to affect the physical properties of the microneedles, the release of the drug, and maybe the overall storage stability of the actual product itself. So clearly the design of our patch and also the geometry of the microneedles will have a big influence on what we can deliver. So if we've got a controlled release tip to our microneedles, or we've got solid drug nanocrystals making up our drug component in there, we probably aren't going to deliver anything that's above the stratum corneum. We really need to get that beneath the stratum corneum. So we need to think, where is the drug or the implant tip positioned on the length of the microneedle, and what percentage of that needle length actually goes beneath the stratum corneum and can actually deposit its cargo. How close can we get the needles without having a bed of nails effect? And then what overall patch size can we make that a person can still easily apply? All of those things will influence the deliverable dose. And then the nature of the drug or the particle or the implantable tip will all affect the duration of delivery if we then peel off the patch after the needles have been deposited beneath the stratum corneum. And as I said, unlike a small water-soluble molecule, if we've got a hydrophobic drug particle, we're probably not delivering anything that's in the base plate, so we shouldn't put any drug into the base plate. We also don't want any sticky drug or polymer residue on the surface of the skin for aesthetic and commercial reasons. Patients aren't going to like it, and therefore it's not going to be something that a company would want to pursue. So the first condition that I think can be treated in this way, and Darren touched on this during his presentation, is HIV, and then AIDS obviously is the disease that HIV causes. And this really was and remains the original pandemic, with more than 33 million people having died to date due to HIV infection. There were approximately 38 million people worldwide living with HIV or AIDS in 2019, and there were actually 150,000 new infections in children in 2019 that year. So it's still very prevalent and still continues to spread. The vast majority of people living with HIV are in low and middle income countries. But the good news is that 21.7 million people in 2019, which is 67%, were actually accessing antiretroviral therapy. And this can mean that people can live a more or less normal life and have a normal lifespan by suppressing viral replication. And actually detectable levels of virus in the blood are very rare in people who continue to take their medicine. And what this means is that in the 20 years up to 2019, new HIV infections actually fell by 39%. People who are properly treated can't actually spread the disease. And deaths fell by 51%, with 15.3 million lives actually saved due to the treatment. Problem is that people get really fed up with taking tablets and capsules every day, especially if they have to take multiple solid dosage forms every day. This is a constant reminder to them that they are unwell, and this can lead to a lack of adherence to treatment. So that's whether people are being treated for HIV or whether they're taking an antiretroviral drug as a pre-exposure prophylactic. Again, that's reminding them that they're engaging in risky behavior, and this can have a big psychological effect on people, leading to discontinuation of treatment. So long-acting injections of poorly soluble or hydrophobic drugs is an alternative. So basically, an injection is given intramuscularly on a monthly or every 2 months basis, and the solid drug nanocrystals slowly dissolve and are absorbed from the microcirculation in the muscle to give therapeutic plasma levels of the drug. So 2 drugs are used if it's for HIV treatment, and a single drug is used for prevention of HIV in high-risk individuals. But really, microneedles would be very advantageous over hypodermic injections in this case, particularly in developing countries where up to 50% of injections given are unsafe due to the lack of healthcare workers. And also, we have big problems with reuse of needles, inappropriate disposal of needles, and needle stick injuries leading to transmission of HIV or hepatitis. A person may also have to, as I mentioned, walk for 2 days to get to a healthcare setting and 2 days to get back. That's 4 days that they're not actually working. And that means they may struggle to feed their families. So if we could have a person getting a box of patches and being able to take them home, then this would be a big advantage. So in terms of formulation of long-acting microarray patches then containing drug nanosuspensions, then we will take our drug as solid drug nanocrystals. So these are basically tiny crystals of the drug, somewhere between 200 and 400 nanometers, although they can be as big as 600 nanometers in diameter. They don't tend to be spherical like polymeric nanoparticles. They tend to look like tiny knives. And basically they're made by essentially grinding up large drug particles, and they're stabilized with surfactants and polymers, and they basically slowly dissolve in the interstitial fluid. So we take that nanosuspension, mix it into a gel at high concentration, and then load that into our mold to form microneedles. We then come along and we form a base plate with another polymer, preferably one that doesn't stick too well to the surface of the skin and doesn't have a strong cohesion with the microneedle bodies, and then a sticky backing layer. So we want the base plate to readily detach upon microneedle dissolution in skin, and then we want the nano-formulated drug to slowly dissolve over a period of a week or even longer than that to sustain plasma levels of the drug. So the first drug that we studied in this area was Janssen's rilpivirine molecule in collaboration with PATH and sponsored by USAID. We basically took their injection, we mixed it with some polymers, PVA and PVP. We made dissolving microneedles and we applied those to the skin of rats. And then we followed the plasma levels and also the vaginal tissue levels over a 56-day period in the rats, compared to rats that had got Janssen's intramuscular injection nanosuspension. The reason we looked at vaginal tissue was that HIV is most commonly transmitted by heterosexual intercourse. So basically what we found over the 56 days was that the microneedles performed a little bit better than the intramuscular injection in terms of the plasma levels, which remained above the therapeutic concentration for rilpivirine over the 56 days in both the plasma and the vaginal tissue. Now, these long-acting nanosuspensions tend to exhibit what is known as flip-flop pharmacokinetics. So what this means is that the overall plasma exposure or area under the plasma concentration time curve is not controlled by elimination of the drug in the way it would be if we administered a water-soluble drug by injection. Instead, it's the slow dissolution phase of the nanocrystals that actually controls the overall pharmacokinetic half-life, if you like, and therefore the duration of effective delivery. And so doing allometric scaling from a rat to a person is not just so easy in this situation, especially when you're using the intramuscular bolus as your comparator. You clearly will give a much higher intramuscular bolus to a person than you will to a rat. And that bolus will tend to either all stay together because it's a series of hydrophobic nanoparticles being put into an aqueous environment, almost like a long sausage in the muscle. or a granuloma will form around that, which has a further controlling release activity. And clearly that bolus will be an awful lot smaller in a rat. And so that makes allometric scaling for microneedles, which in theory should form bolus sizes that are identical in rats or people, providing insertion depth is similar. It makes it challenging, but we've had a go at it in some work with pharmacokinetic modeling colleagues in the University of Liverpool. And based on this, we were estimating a patch size for 7 days human treatment at being somewhere between 25 and 30 square centimeters. So somewhere close to the size of a conventional transdermal patch rather than a conventional microneedle vaccine patch. The next molecule we looked at was etravirine, and one of the things that was in our minds here was that formulating nanosuspensions requires a lot of energy to grind up large crystals into small crystals, and this can be expensive in the pharmaceutical industry. So what we wanted to do was see, well, can we just take the etravirine powder straight out of the bottle, mix it with a gel and form microneedles, and compare that to an etravirine nanosuspension that we've made. So we made both types of microneedles, and what you can actually see at the top there are the microneedles containing the powder, so they're less homogeneous than the microneedles containing the nanosuspension, which you can see these little flecked crystals in the microneedles. So there's no proprietary long-acting nanosuspension of etravirine. So our comparator in the rats was an intravenous injection of etravirine, and we then applied the different types of microneedles. And what we found was that the intravenous injection gave a very high initial plasma concentration, but within about 10 days, this had really started to fall. Whereas with both microneedle formulations, we were still above therapeutic plasma levels a month after application. And what this shows is that you can control the release long after the microneedle patch is removed, because like the rilpivirine patch, we remove this patch from the rat skin after 24 hours. But because we're depositing these poorly soluble drugs, whether in sort of microparticle form or nanocrystal form, in the viable skin layers, and the drug then slowly dissolves. So it seems that it doesn't really matter what the actual particle size is here. We still get a sustained release effect over the month for etravirine. So again, quite interesting for this HIV molecule. The next drug we looked at was Gilead's Bictegravir. And again, we used microneedles that were made from either a nanosuspension that we engineered ourselves, or just the drug powder loaded into dissolving polyvinyl alcohol, polyvinyl pyrrolidone microneedles. We applied them to the skin of rats, and then we gave an IM injection of the nanosuspension that we'd made ourselves. And what we found was that the microneedles actually performed better than the intramuscular injection, but the relative bioavailability of the microneedles was probably a quarter of what it was for the intramuscular injection. So we're still not delivering all of the drug, but we were able to maintain a therapeutic plasma level for at least 3 weeks from a single microneedle application. So very similar in terms of performance to the intramuscular injection, but just with the caveat that we're not delivering all of the drug. Some of the drug will actually remain on the surface of the skin. So what we can do is we can improve the efficiency by pushing the drug more to the tips of the microneedles, and that will improve the efficiency of delivery, but you won't deliver any more drug per unit area. And so this won't reduce your patch size from what it is at the minute. And again, we think with some allometric scaling for both Bictegravir and etravirine, the patch size is probably going to be somewhere in about 30 square centimeters, but for weekly delivery rather than monthly delivery. And, you might think, well, a person would need to apply a patch every week rather than getting an injection every month. That might be inconvenient. I mean, in my experience of working in community pharmacy, I think a patient would remember to apply a patch once a week on a Monday morning, more so than they'd remember to apply a patch once a month, the first day of every month. So I think that I would actually take that as a positive that the patch would be applied on a weekly rather than a monthly basis. The next example is CABOTEGRAVIR. So again, this is a project funded by USAID and in collaboration with PATH. This is ViiV Healthcare's molecule, CABOTEGRAVIR. So here we gave their long-acting nanocrystal injection intramuscularly or intradermally, and we compared that to the pharmacokinetics of 3 different types of long-acting microneedle patch. So the first one, where we just took the long-acting injection and mixed it with PVA and PVP to make the microneedles. Then we took the powder form of the free acid from which the injection is ultimately made, and we made microneedles from that. And then finally, we took the more water-soluble sodium salt in just micronized powder form and made microneedles. The critical point was that the microneedle plasma levels were less than they were for intramuscular or intradermal injection. But over the 28 days, all 3 microneedle formulations yielded plasma concentration that were above the therapeutic plasma level for CABOTEGRAVIR, which is 4 times the protein-adjusted inhibitory concentration, 90%, of about 664 nanograms per mil. Very interestingly, the more soluble sodium salt microneedle system actually performed just as well as the intramuscular injection and intradermal injection over 7 days. And I think that this was particularly notable, that by increasing the solubility of the drug a little bit, we were able to match the performance of the injection for the first 7 days. But even after 28 days in the rat, we were still above the therapeutic plasma level. Again, we think that weekly dosing in people would be the most realistic, and we're not going to have an enormous patch size for that. So something similar to the previous patch sizes. So single agent HIV drugs can be used for prevention, but not treatment. So for treatment, you need 2 molecules together. So again, this is in collaboration with PATH, with support from ViiV and Janssen, and funded by NIH in the United States. We've delivered both CABOTEGRAVIR and rilpivirine to the rats in this case. Basically, we applied microneedle patches individually containing both drugs to the back of the rats. And we compared this to intramuscular or intradermal injection of the drug combination in the same rats. And we looked at 2 different microneedle geometries. So for rilpivirine, what we found was actually that we had therapeutic plasma levels beyond 60 days, regardless of the microneedle design. In this case, with rilpivirine, we're only aiming for the inhibitory concentration 90%, which is typically the known therapeutic plasma concentration of rilpivirine. After 60 days, we were still above therapeutic plasma levels, and we outperformed the injection in this case. And then in the rats, again, the intramuscular and intradermal injection, we're seeing higher plasma levels than we are with the microneedles, but the microneedles still perform well. And regardless of the microneedle design, we still end up with therapeutic plasma levels after 28 days. So this looks very promising. We can deliver 2 drugs, so treatment would actually be possible. It's not just prevention. We then wanted to mimic the real life situation where patches would be repeatedly applied. So we looked at applying patches every 14 days after either an intramuscular loading dose of CABOTEGRAVIR and rilpivirine, or a loading dose that just involved applying the microneedle patches. And what we found was that after 14 days with rilpivirine, we actually were matching the cohort that had the intramuscular bolus just with microneedles on their own. With CABOTEGRAVIR, it took a little longer, but by 28 days, we were matching that. So you could have a situation where maybe you want to reduce the patch size, and you have a person go into a healthcare setting, instead of every month, maybe every year or every 6 months, getting an intramuscular injection. And at that point, they get their normal physical checkup, and they get a box of patches home with them to essentially top up the drug that was being cleared. So again, this is something that we find very encouraging, and we have attracted a lot of interest from companies in this area of long-acting delivery of a range of different molecules, not just HIV type molecules. All the drugs I've talked about so far in this long-acting space have been hydrophobic, but there are some HIV drugs that actually are hydrophilic. So we can't just hope that putting a high amount of drug into the microneedles is going to do the trick. So with Tenofovir Alafenamide, so again with support from Gilead, we looked at two approaches. So one, we just loaded the drug as a powder into the dissolving microneedles made from PVA and PVP. And then in another iteration, we made the tips of the microneedles from PLGA, containing the more hydrophilic molecule. So again, we compared this to intramuscular injection in rats, and what we found was the intramuscular injection gave a high peak, but then rapidly fell off. This was similar to the dissolving microneedles because the drug is actually quite water soluble. But the implantable microneedle systems with the PLGA tips give plasma levels that remained higher for a bit longer. And we can't quite see it so easily here on this graph, but if you magnify that out, we see that actually for the injection and even the dissolving microneedles, we still had therapeutic plasma levels of Tenofovir Alafenamide as measured through the tenofovir active drug levels that were still active after 7 days. So again, we think a patch applied every 7 days could be suitable even for a water-soluble drug like tenofovir, but you probably would be best slowing down its dissolution by using a biodegradable polymer in this case. So I think that there are lots of options for HIV long-acting treatment, and there are a range of other diseases that could also be treated using similar techniques. Another condition, though, that where microneedles could actually have a big role is in lymphatic filariasis. So this is caused by the bite of a fly that basically transmits a parasite into the body, and the parasite is essentially a worm with a bacteria in its gut. And that finds its way to the lymphatic system and causes inflammation. This causes damage to the lymphatic system, and this causes the swelling and disfigurement associated with what's often crudely known as elephantiasis. Over 120 million people are infected, with about 40 million people disfigured and incapacitated by the disease. You can treat the microfilariae and bacteria in the bloodstream by taking the drugs orally, but once you have a lymphatic infection, you have poor transmission of the drugs into the lymphatic system. And as a result of that, you have a big issue with actually managing the condition. So one way we could target the lymphatic system is by putting in particles that are less than 100 nanometers in diameter into the skin interstitial fluid, because skin is high hydrodynamic pressure. We also have a high density of lymphatic vessels. And as a result of that, we could have a good way of targeting the lymphatic system. So what we did here was we took a number of relevant anthelmintic and antibiotic drugs. We loaded them into solid lipid nanoparticles, which we then put into the microneedles, applied them to the skin of rats, and then sacrificed the rats at different times and excised the lymphatic system. And what we found was that the lymphatic concentrations or concentration time profiles for doxycycline, albendazole sulfoxide, and albendazole sulfone, we were basically getting 7 times more delivery into the lymphatic system using the microneedles than we were when we gave the same drugs orally. In fact, the only drug that we saw that trend reverse was for albendazole sulfone, and actually this was kind of a test because we knew this would happen. Albendazole sulfone is a metabolite of albendazole that is only formed during first pass metabolism through the liver. So it didn't surprise us that oral delivery led to more of this drug. So we think that lymphatic targeting can have a lot of potential, not only for infectious diseases like lymphatic filariasis, but also for a number of different immune conditions, autoimmune diseases, metastases of cancers, and also primary cancers of the lymphatic system, where we could potentially reduce side effects to the whole body. by targeting the lymphatic system using microneedles. So the next technology that has long-acting potential doesn't involve depositing solid drugs or implants in the skin. These are hydrogel-forming microneedles, which swell in the skin, of course, and form a continuous unblockable conduit between a patch-type drug reservoir and the dermal microcirculation, and drug delivery will continue as long as the patch is left in place. And in some work with industrial collaborators, we've been able to leave the patch in place in pigs for 5 days and continually deliver drug at a therapeutic level for that entire 5 days, but crucially remove the patch with all of the microneedles intact after the 5 days. So we don't deposit polymer in the skin, but we've been able to do a long-acting delivery while the patch has been in place. And this can be suitable for weekly administration, for example, of water-soluble molecules, including large molecules. So one very quick example is the antibiotic metronidazole. Here we applied the microneedles to the back of the rats for 2 days, and we continually maintained plasma levels that were therapeutic for the full 48 hours. And if we had extended the experiment for longer than that, we probably could maintain the therapeutic plasma levels for even longer than that, because we can put as much drug as we want into the drug reservoir that sits on top of the microneedles. We're not limited by what we can load into the microneedles in that case. So this can be promising. Another example then is delivery of a very large molecule, Avastin or bevacizumab. In vitro, across excised pig skin, we were delivering 50% of the drug loading in 24 hours. And we do believe we could deliver even more if we kept the patch in place for longer than that. And in the in vivo setup, we compared microneedle delivery to in vivo delivery. And for a single microneedle application, we were seeing week-long plateau, whether we used high dose or low dose microneedles. But notably, the plasma levels were lower than they were for the intravenous injection of the same drug. And this is because of the size of bevacizumab. It's 250 kilodaltons. Anything greater than 12 or 16 kilodaltons isn't going to be easily absorbed by the dermal microcirculation. So it's going to go into the lymphatic system first. So this could be a way of extending the lifetime in the plasma or in the body of therapeutic antibodies for even longer than if you give them intravenously. But also it could be a way of targeting these therapeutic antibodies directly to the lymphatic system. And we could get quite an important range of therapeutic uses in that way, while also minimizing whole body side effects. So this is something that we continue to study, also in collaboration with some industrial colleagues. So I've mentioned a lot about large patches, and it's important that people can apply large patches. So what we did was we got volunteers to apply a large 16 square centimeter microneedle patch and also a small 1 square centimeter microneedle patch, using a pressure responsive film that would change color if and only if the correct pressure had been applied at every point on the area of the large patch. So if a little corner didn't change color, the person knew to apply pressure there again. We came along with optical coherence tomography, the optical analog of ultrasound, and basically measured the depth of insertion and the width of the pores created at various different points on the large patch. And also then we looked at the small patch in the same volunteers, and we couldn't find any significant difference between insertion depth or the width of the pore created. These people had used this pressure responsive film that had also been counselled by a pharmacist and read a patient information leaflet, but no special applicator device was required other than the pressure responsive film. So we've mentioned a lot then about microneedles for drug delivery being used more than once. So they might be used daily, weekly, or monthly. So we need to show that repeat application of microneedles is safe. So we took some nude but completely immunocompetent mice. We applied microneedles to their skin once a week for 5 weeks. And then we came along and looked at them at the end and found that skin appearance was unchanged relative to control. We couldn't see any evidence of polymer accumulation underneath the skin. And when we measured transepidermal water loss as a measure of skin barrier function, we found no change relative to control. We then looked at immune response, and what we found was whether we used dissolving Gantrez S97 microneedles, whether we used dissolving polyvinyl prolidone microneedles or swellable hydrogel-forming microneedles, there was no measurable increase in immunoglobulin G relative to control, regardless of whether we used male or female mice or what density of array of microneedles we used. And we didn't see any significant difference whether the microneedles were dissolving in the skin or whether they were swelling in skin. So repeat application of microneedles themselves doesn't seem to induce any strong immunity in this population of mice. We then wanted to see, was there any action on inflammation? So we measured interleukin-1 alpha. And again, needle density had no detectable effect. And even the low pH of the Gantrez S97 formulation had no significant impact relative to control. So again, this looks promising. We then looked at infection by measuring C-reactive protein. And again, there was no measurable increase in this relative to control. We didn't see any increase in body temperature in any of the animals, and there was no significant weight change. So again, application of microneedles on a repeated basis seems very safe. We talked before in the conference about the microbiological status of microneedles, and we made various different microneedles, both dissolving and hydrogel forming microneedles, and we did sterility tests according to the British Pharmacopoeia, and we could never detect any viable microorganisms after the microneedles had dried. And we even deliberately spiked the microneedles with Staphylococcus aureus, Pseudomonas aeruginosa, or Candida albicans, and there was no survivors. And hydrogel forming microneedles actually exhibited anti-adherent properties, which stands to sense they're a hydrogel, so they become slippery and things don't stick to them. And when we measured endotoxin levels, they were below all pharmacopoeia limits, even for injection into the blood system or into the lymphatics. And then when we used a radio-labeled lawn of bacteria or fungi on excised skin, and we applied the microneedles, we basically found that there was significantly lower permeation of microorganisms through holes in skin compared to hypodermic syringe puncture. So all this looks very promising, but I take Michael's point that regulators may still require a sterile product. We wanted to show that microneedles could be terminally sterilized. And what we found for dissolving microneedles, of course, was that we had a change in drug delivery kinetics, but for hydrogel forming microneedles, probably because they have already been cross-linked before we hit them with 60 kilograys, that their swelling properties and the delivery properties of both small and large molecules were unaffected. And that's in contrast to dissolving microneedles where we did see a reduction, but I haven't shown that data here. And then finally, in terms of repeat application of hydrogel forming microneedles, we did this in human volunteers. We applied microneedles to their skin every day for a week. And then we looked at the skin, and we saw erythema, of course, when we removed the microneedles. This all was recovered within 24 hours. Similarly, transepidermal water loss, it was raised after we removed the patches and then returned to normal when we came and measured the next day. And then we measured various different biomarkers at the end of the week-long period: immunoglobulin E for allergy, C-reactive protein for infection, immunoglobulin G for an immune response, and then tumor necrosis factor alpha for inflammation. And they were all unchanged relative to control. So again, repeat application of microneedles themselves as a platform seems to be safe in people as well. So I think that there are great potential benefits from microneedles for delivery of high-dose controlled release therapeutics, and we can deliver more than one drug from the same platform. And we can control and tailor administration even long after the patch is actually removed. And this is potential to address a range of global healthcare challenges. So not only HIV or antibiotic-resistant infections or lymphatic filariasis, we could be treating tuberculosis, schizophrenia, we could be providing long-acting contraception, and a range of other applications. And actually, personally speaking, I think that there have been great strides made recently, and we'll see from Lisa and Sebastian a little bit about this later on, no doubt, that the manufacturers have made a huge impact in recent years in terms of development. And we've also seen through Path Centre of Excellence a big push on regulatory development and understanding critical quality attributes and starting to develop suitable quality control tests. Our own work has seen significant investment from Big Pharma, and I think that Big Pharma are realizing now that the real potential of microneedle systems. Our own work has recently received a major program of funding from the Engineering and Physical Sciences Research Council in the UK, worth several $1 million. And the purpose here is to de-risk the delivery platforms in accordance with regulatory advice and basically optimize the design of microneedles. And this will all culminate in a bridging program that would be applicable to any drug to be delivered. So basically it would show that the microneedle platforms are safe. We could hand that to any potential commercial partner, and then they can use a very defined bridging study to show that their drug, when delivered with that already proven to be safe platform, is not going to cause anything detrimental to patients. And I think that this will then inevitably lead on to further clinical trials, which will support ultimate commercialization and patient benefit. And I really am optimistic now about the impact that microneedles could have in improving therapeutic outcomes for patients right across the world. So that concludes my presentation. All that remains is to thank the sponsors of our work, the members of my research group, and also to thank you for your kind attention. Thank you.
Thank you.